Now let's move our conversation from the membrane-bound organelles to the non-membrane-bound organelles, which is really a fancy term for the elements that comprise the cytoskeleton. Note that the cytoskeleton consists of three different elements. The smallest of them are microfilaments, consisting exclusively of actin. Kind of intermediate in size, hence the name intermediate filaments, are tissue-specific filaments that consist of a number of different proteins that provide a structural integrity to cells in a wide variety of organs. Lastly, the largest of the cytoskeleton elements will be the microtubules, and we'll see a variety of locations in which we find them. First, let's start with microfilaments. Microfilaments consist exclusively of actin proteins that basically are globular monomers of G-actin that have polymerized to form helical filaments of F-actin. The actin polymerization is energy dependent. They typically are about 7 nanometers in diameter, and they have a distinct polarity. The barbed end is the site of polymerization, and the pointed end depolymerization. And treadmilling is in balance and activity at both ends. And one of the reasons this is important is that microfilaments convey a motility to cells, particularly cells that are actively moving in and out of the bloodstream, such as white blood cells. But in addition, microfilaments will form a contractile ring that will be used in cytokinesis of cells undergoing mitosis or meiosis. And lastly, microfilaments, actin proteins, will form the core of a cell surface specialization known as a microvillus. Next, what about intermediate filaments? They are consisting of tissue-specific proteins, and overall they convey a structural integrity to a particular cell. And there are a couple of different different types of intermediate filaments. Type 1 consists of keratins, and keratins basically are found in all epithelial cells, whether they are ectoderm, mesoderm, or endoderm derived in terms of embryology. Type 2 have a fair diversity of them. Desmin is a type 2 intermediate filament that's found in virtually all muscle cells, particularly skeletal muscle cells, cardiac muscle cells, and at the very least GI tract smooth muscle cells. Vimentin, another type 2 intermediate filament, is found in fibroblasts, endothelial cells, and blood vessel or vascular smooth muscle. It forms what is known as glial fibrillary acidic protein, which is found in glial cells such as astrocytes and most Schwann cells, and peripherin, which is an intermediate filament that maintains the structural integrity of axons in the peripheral nervous system. Type 3 are intermediate filaments that are found actually forming structural supports in the bodies and dendrites of neurons. And type 4 intermediate filaments form lamins that form a meshwork inside the nuclear envelope of all cells. Finally, microtubules. Microtubules are in essence hollow tubes. They are the largest of the cytoskeletal elements, and like microfilaments, they are undergoing continuous assembly and disassembly because one of their functions is intercellular transport of vesicles, molecules, and they're particularly important in the nervous system because they are the subcellular organelle that's used in axonal transport in both the anterograde and the retrograde direction. Transport, regardless of where it is an energy-requiring process, and dynein drives what's known as retrograde transport. Kinesin drives the component of anterograde transport in neurons as well as any other cell type that utilizes microtubules. So, in addition to providing vesicular and molecular transport inside cells, microtubules are the cytoskeletal element that are found in true cilia and flagella, and as such, because these are motile structures, they utilize dynein to maintain the beating nature of true cilia, and they all obviously are found in the tails of sperm cells, allowing them motility as well. And then finally, not the least of which, is microtubules form the mitotic spindle, which is going to help during chromosome separation during mitosis and meiosis. All right, One of the things that affects intermediate filaments, which is particularly relevant in the nervous system, is Alzheimer's dementia. But we also see it in cirrhotic liver diseases as well, that basically are caused by initially changes in the intermediate filaments. Colchicine prevents microtubule polymerization and is used to prevent neutrophil migration in gout. And there also are elements, vinblastine and vincristine, that are used in cancer therapy because they inhibit the formation of the mitotic spindle of dividing cancer cells.